This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 122, recorded February 25th, 2011. Hi, everybody. I'm Vincent Racaniello, and it's time for TWiV, where we tell you all about viruses. Joining me today, starting over in Fort Lee, New Jersey, the venerable Dickson Despommier.
Hey, Vince. Hey, guys.
Do you like the venerable title?
Not bad. Vulnerable would be worse.
How is everything in Fort Lee?
It's raining.
Yeah, I know. I can see you across the river here. It's raining here, too, in New York City.
Yep.
Also joining us from Western Massachusetts is Alan Dove.
Good to be here, and it's raining here, too.
Raining as well? Good
yeah.
Good to hear you again. Haven't heard you for a few weeks.
Yeah, good to be back.
Did you miss TWiV?
Yeah, I did.
Yeah, I missed you, too.
There was a hole in my Friday afternoons.
And then moving down the coast to north central Florida, Rich Condit. Hey, Rich.
Hi, fellas.
Hello, Rich.
How's everybody?
Feeling good.
Let me guess, it's not raining there, right?
No, it is not raining. We have partly cloudy skies, 83 degrees.
Nice.
Oh,
nice.
It's springtime here.
Sounds
Wow.
great.
And then we'll move back up the coast to the same state as Alan's, in fact. Somewhere outside of Boston, I guess, is Welkin Johnson. Hey, welcome back, Welkin.
Glad to be back.
What a harmonious thing to say. Welcome back, Welkin. That's sort of a tongue twister, isn't it?
I've never heard that before.
No? Well, maybe no one asked you back again. I'm just kidding. Welkin is a professor in the Department of Microbiology and Molecular Genetics at Harvard Medical School. And we had you on a while ago to talk about endogenous retroviruses.
That's right.
And you are in the department that used to be chaired by Bernie Fields.
That's right, yeah.
Who was a giant in viral pathogenesis, of course. You're also a associate blogger at Elio Schechter's blog, Small Things Considered. Are you having fun there?
Oh yeah, I think I don't contribute as much as I should, but it's a great blog.
Yeah, well, you have to do other things with your career right now, right?
Other things to do with my career, and of course, we're always trying to catch up with TWiV.
Yeah, we keep you busy, huh? Today, we are going to talk about what you do.
Oh, and thank you for not asking me about the weather here.
I figured it was the same as Alan's.
We don't speak of it.
Yeah, I have some fascination with the weather. I don't know why. I could just cut it out from the banter in the beginning. We'll think of something else to talk about. Before we start talking about you and your work and trims and sieves and HIVs, all of which will become clear in a moment, I just wanted to point out the death today of a virologist, and that is Edwin Kilbourne, MD, who was retired, but for many years was chairman of the Department of Microbiology at Mount Sinai, which is where I got my PhD, so I knew Dr. Kilbourne, and he was a quite significant flu vaccine expert. He is perhaps best known for encouraging the nationwide immunization against swine influenza in 1976. And he had a long, illustrious career. He was, in fact, responsible for the practice of making high-yield recombinants of current influenza strains with a strain that grows well in eggs so that that could be used for vaccine development. I remember he had a technician who used to get the selected strain every year, and she would make recombinants in eggs, and that would be provided to the vaccine manufacturers.
Wow.
And I also should point out that I went to college with Edwin's son, who is also named Edwin. And for a long time, Edwin Jr., he would be mad to hear that, I think, worked at the CDC. He was also an MD. And now I believe he's in the private sector, as they say. But Dr. Kilbourne Sr. Is responsible for my career in virology to a large extent. And we'll leave it at that because someday someone can ask about that, and I'll tell the story. So
I can't ask now.
No, I'm not going to do it now. I don't want to take time away from Welkin. This is Welkin's day.
Go ahead.
And we'll do it another time. And,
Well, you better put it on a list somewhere.
A list. Yeah, I have a lot of those.
I'll bet.
And I just wanted to note the passing of Edwin Kilborn. You know, I had really planned. He was retired, living up. I think in
Connecticut.
Was it Connecticut?
Yeah.
Okay. I had planned to go with a mic and interview him, you know, and play it on TWiV. And I didn't get around to it.
Well, there's one of those lessons, right?
You should just do it.
Right.
In fact, I asked his son, hey, do you think your father would like to talk to me about virus? He said, oh, I'd love it. He's really on top of it. But now I can't do it. So that is my regret. The lesson is if you have an idea, just do it. Follow through. Okay?
All right.
All right, guys?
Depends on what the
I'll do
idea.
that.
Depends on what the idea is.
Yeah, well, if it's
I don't have any of those kinds of ideas, Alan.
Can I make people in my lab do the idea?
well, I gave a seminar in Seattle last week, and I told a story, and I said the moral is if you have an idea for an experiment, you should do it. Don't let anyone talk you out of it, because it was a story where someone had talked me out of an experiment. And then someone else did the experiment, and it was a great result. And to this day, I wish I had done the experiment. Right. So, I don't know about life, but in science, do the experiments you want. Even if your PI doesn't like it, just do it. Because
Absolutely.
if it doesn't work,
You don't have to tell anybody.
You don't tell anybody. And if it works, then you look really good. All right, that's that. Now, the last time Welkin was here, we talked about endogenous retroviruses, but we never got to what you do. And what you do is pretty cool because you work with restriction of SIV, simian immunodeficiency viruses. And I want you to tell us about that today. And you had a good idea to tell this as a historical tale. You call it the other AIDS epidemic.
Yeah, that's right.
Simian immunodeficiency virus. So take it away. And we'll interrupt you, by the way, if you don't mind, with questions.
So the history all predates my getting into this field. It's a lot of work by a lot of interesting people. That all sort of took place before I started working on it. And so in my mind, the story starts with outbreaks of opportunistic infections and immunodeficiency that was occurring throughout the 1970s in colonies of Asian macaques in U.S. biomedical research centers. So at primate centers in the United States, these Asian monkeys were getting wiped out by this disease.
So this predated HIV, right? AIDS.
Yeah.
Wow.
So this was through the 1970s. And so what, as you pointed out, around 79 or 80, a very similar thing started to crop up in humans in L.A. and in New York and San Francisco. And so if you fast forward a couple of years, this is all before I was born, of course. In 1982, 1983, they identified HIV-1 was identified as the etiological agent of AIDS in humans. And it was very shortly after that, 1985, I think, when researchers at the primate centers, including the primate center where I work, people like Muthaya Daniel, Ron DeRozier, and Norm Letvin, cloned and sequenced the virus that was causing this disease in monkeys. And when they did that, and looked at the sequence, the genome arrangement and the sequence were very similar to HIV-1. It looked like a very close cousin of HIV-1. And so that virus came to be called simian immunodeficiency virus. That was the first SIV that was discovered. And of course now there's 40 or more of them that are known in wild monkeys in Africa. But that was the first SIV. And so that story alone, for some reason, isn't widely known, even in the HIV/AIDS field. I think most people don't know that the origin of SIV is unclear in Asian macaques. It wasn't an intentional experimental model. It's something that just eerily enough just dropped out of the sky almost at the same time that the same disease was emerging in humans. So you have an emerging infectious disease in humans, and the animal model for that disease appears in biomedical research centers just by chance at the same time.
Now, was that a byproduct of the Asian macaques being kept with other primates that might have been able to harbor SIV without symptoms?
You know, that's a great question. So nobody really knows what happens, but if you talk to Christiana Petri, who's in Pittsburgh now, but he used to be in Tulane. He's actually got a paper that you can look up where he's put together a bunch of sort of looked at colony records and some of the work they've done looking at samples. And actually, he has a very specific hypothesis that Carlton Gajicek, so he's the Nobel laureate that figured out that Kuru was an infectious disease.
Right.
That Gajicek was actually in the 60s and 70s, before he knew what caused Kuru, was trying to induce in the animal model of it. And so apparently what he was doing was taking tissue samples from one species and injecting them into another to try to and so if that's true, that would be a very likely way that it could have happened, because African sooty mangabees and Asian macaques were both species that were present in the primate center. So it's very, I think you'll have to talk to Christian about it, but he makes a very compelling case that that's probably what was going on.
The monkeys in the facilities were all imported, right? Or they weren't bred here?
They started by being imported. Mm-hmm. The import was, I forget when, but it's been a while since we've been able to import them. So for a decade or more now, I think they've actually been bred.
But it could have been imported a long time ago and not detected for a while, perhaps?
Well, it was probably imported in sooty mangabees from Africa.
Okay.
And then during the 60s here in the States, somehow the virus that was not causing disease but circulating in mangabees wound up in the Asian macaque colonies, possibly as an unintentional consequence of these experiments.
So is it still present in U.S. colonies?
Is SIV? Yes. There's sooty mangabee colonies that are that still have SIV in them.
And how is it transmitted among macaques?
I don't. So now in the macaques, it's not being transmitted other than by experimental transmission. But I imagine at the time it was probably as they become juveniles, I think they start fighting and scrapping with one another. Mm-hmm. I'm embarrassed to say I don't know exactly.
You could just say it's sexually transmitted. We'd buy it.
Well, I don't know. I actually don't know if that's the primary or the only reason that it happens in nature. I think it may be when they start fighting. Mm-hmm. But I don't know that for a fact.
Now, so coincidentally, at the same time, you have a primate virus leaping from primates probably in Africa to humans, and that's HIV. So when you trace these trees back, how parallel are they?
So the work in HIV, I'm not going to get the dates exactly right, but the people who do that, like Betty Korber, I think, believe that the transmission occurred early in the 20th century, whereas the transmission of SIV to macaques almost certainly happened in this window of time between 1960 and, say, 1972.
Right.
And so what would have been hypothetically at least, what are the circumstances of the transmission of HIV from apes to humans?
What are the circumstances?
Yeah, where would that have happened? I mean, is this a hunting thing or?
Yeah, Beatrice Hahn, I think, talks about this quite a bit, and I think she puts forward the theory that it can happen with hunting and butchering of monkey meat, which is sold in markets in Africa. And I believe that's her favorite hypothesis.
All right.
So the SIV in the macaques then was an experimental transmission, but it wasn't an intentional one. There were no known primate lentiviruses at the time that it happened.
Okay.
So then, should I go on with the story?
Sure.
Yeah,
sure.
Absolutely.
No, we're done. Okay. Bye, everybody.
I want to hear more.
I'm one of those people that can go off on tangents forever.
It's okay.
So,
we don't know.
You're in good company.
No, you join the group. That's right, exactly.
so SIV was discovered, and I guess one of the immediate questions was why is it that these SIVs can replicate in monkeys and monkey cells and HIV-1 can't? Because what everybody was trying to do at the time, or a lot of people were trying to do, is come up with an animal model for HIV-1 infection. And no one's ever successfully done that, although recently there have been some progress. And so SIV infection of macaques has then been the animal model for 25 years now. And one of the early things that was done, and this starts to get to the history of TRIM5, is that there were some researchers in Japan. There was Riri Shibata and Hayami and Adachi, and here Bill Hazeltine and Joe Sadrowski and others were taking the SIV and HIV genomes, and they were mixing and matching pieces of them. So they were making chimeric viruses and asking whether or not those could grow in monkey cells. And what they found universally was that you could get chimeras to grow in monkey cells as long as the 5' half of the viral genome was derived from SIV. The rest of it could be HIV, but the 5' half had to be SIV. And in retroviruses, the 5' half, the bulk of the 5' half is the GAG gene, which is the structural gene for retroviruses. And GAG encodes the capsid protein. So capsid is the protein that multimerizes, so 1,000 or 2,000 capsid monomers multimerize and form the protein core that surrounds the retroviral genome. And so I think it was probably recognized early on then that whatever was blocking HIV-1 from infecting monkey cells, it had something to do with the HIV-1. The virus side of the story was the viral GAG gene and probably had something to do with the capsid protein. And so I think that brings the story up to the mid-1990s. And then there was a postdoc, I think he was a postdoc named Wolfgang Hoffmann, who I think sort of shifted the field at this point, because what Wolfgang was working in Joe Sadrowski's lab at Harvard, and Wolfgang published what was just a descriptive paper, but it was an interesting one because what he did is I guess he went to the ATCC catalog or something, and he bought a whole bucket load of different cell lines representing a whole bunch of different primate species, so old world monkeys, apes, New World monkeys, and a few other species thrown in for good measure, rabbits and cows and mice. And he did a simple experiment, although it was probably a lot of work. He did single-cycle infectivity assays on all those cell lines in parallel with HIV-1 and with SIV. And what he found across the board was that blocks involving the capsid protein were fairly ubiquitous among primates and could even be found in some of the other species. Sometimes the block was to HIV-1, sometimes the block was to SIV, and some cells, both viruses were blocked. But it seemed like the same phenomenon was going on in multiple different species. So I think that probably came out in the late 1990s.
Now, did he actually narrow that down to the capsid protein as well?
I don't think he did.
It was just replication is blocked.
Yeah, it was early replication. So the way he did the experiment, I think they were GFP transducing viruses. So what he was measuring was a block in the early half of the viral replication cycle.
Because he got no GFP.
Right, so somewhere between entry and integration.
Okay.
So all the cell lines were susceptible to entry?
So the viruses were all pseudotyped, I think, with VSVG protein.
Nice.
So he used a heterologous envelope protein so that he didn't have to worry about whether or not the actual receptor was there.
Gotcha.
So actually, I guess the experiment actually ruled out that entry was the block. Mm-hmm. It said it was somewhere between entry and gene expression. And so I don't know if other people did similar things, but I remember Wolfgang's paper as being one that sort of sparked a lot of enthusiasm. And I think it's probably at that point when other labs started to get interested. And so Ned Landau and Greg Towers, Paul Benash and Theodora Hatziano, who are there in New York City, and probably some others whose names I'm not thinking of, started to really look at this. And so there was a series of papers that came out between 1999 and the 2002, 2003, where the block itself was very well characterized. They came to understand that it involved the capsid protein. It was a saturable block. If you put in enough virus, you could overwhelm it and get virus in. And different things like that, but nobody up to that point, still nobody had actually identified what the actual factor was that was causing the block. And I think a lot of people were probably, beyond the people that I've mentioned, there's probably a lot of us that were trying and failed utterly. And then in 2004, graduate student in Joe Sadrowski's lab named Matt Stremlau did a screen. And it sounds like an easy screen. I think after the paper came out, a lot of people probably either wondered why they didn't try it, or they tried it and wondered why it didn't work for them. But what Matt did is he took a cDNA, rhesus monkey cDNA library, and he shot this into human cells, I think HeLa cells probably. I don't remember. And so some human cell line that was normally susceptible to HIV-1 infection. So he put this library of rhesus genes in, and then he infected the cells with a high MOI of HIV-1 with carrying green fluorescence protein. And what he would do then is sort out all the infected cells, and the cells that were left behind, the ones that were not green, were the ones that either hadn't got infected or somehow had gained resistance to HIV-1 infection. And he did a couple of iterations of that, and then in the end what he had was a population of cells that had a high frequency of a cDNA encoding the gene, the monkey homologue of a gene called TRIM5. And so that's how, and the protein that gene expressed was the alpha isoform, so it's called TRIM5 alpha. And so Matt's paper came out, I think, in early 2004, and that just touched off a revolution at that point.
TRIM stands for?
Tripartite motif, I think. So the gene was already named, I should say. So TRIM5 alpha, I hate the name because it sounds like a diet aid or something.
That's right.
And Matt and Joe didn't name it. The protein was already known, just nobody knew what it did, and so it already had that name.
There are a lot of trims in the cell, right? There are up over 20 some, I think.
Oh, there's almost 100.
100, yeah.
So it's a big family of genes, and people are really only starting to work out what they do. I think it's still a very small percentage that the functions have been worked out, and TRIM5 is one of them.
Are they associated with an organelle?
I'm sorry?
Are they associated with any intracellular organelles?
I don't think that TRIM5 alpha is. What a lot of them do, if you overexpress them in cells, is they form these large agrosomes or cytoplasmic bodies. So sometimes the older literature calls them cytoplasmic body proteins, but I don't know if that's an artifact of overexpressing them or if that's what they really do. I don't know.
I remember those experiments because at the time Jeremy Luban was here.
Yeah.
And they were trying to identify a restriction factor in a different kind of cell than... So Joe was using rhesus cells, right? Rhesus cDNA libraries.
That's right.
I think Jeremy was looking in the owl monkey, something like that, and he found TRIM fused to cyclophilin.
Yeah. So
TRIM-CYP, right?
Yeah. It was very shortly after Matt's paper that... Jeremy and is it David Saya, I think is the student's name. Yeah, they were looking at owl monkey cells, and they knew. I guess. Do you think Jeremy would mind if we mention his work?
It's published.
Because that's kind of a neat story. You know, Jeremy Luban had discovered, when he worked for Steve Goff, had discovered the interaction between the viral gag protein and a cellular factor called cyclophilin A.
Right.
It's not clear to me that we know what cyclophilin A does even now. I think it's been 15 years or something. But what they noticed in owl monkey cells is that if you treated those cells with inhibitors of cyclophilin A, HIV-1 could infect the cells. So normally they were resistant to HIV-1. If they treated with these inhibitors, they became susceptible to HIV-1. Mm-hmm. And they also figured out that it had nothing to do with the cellular cyclophilin A that was expressed in the owl monkey cells. And what they pulled out was a gene, was the TRIM5 gene, but in owl monkeys it turns out that half of the TRIM5 gene, the C-terminal half, has been fused with another gene that encodes a cyclophilin A domain. And so owl monkey cells make this TRIM-CYP-A, or cyclophilin A fusion protein, and because cyclophilin A can bind to HIV capsid, it targets the TRIM5 gene to HIV-1. So it's a TRIM5. It's an example of TRIM5 evolving a new exon, essentially, an exon encoding cyclophilin A.
Right. That fusion is via splicing, right? a wonderful example of modularization of genes.
Yes, yeah.
Jeremy was here a few weeks ago, and he has a new idea for what TRiM5 is doing.
That's right.
And he came up to your place, or Worcester at least, I guess, after he was here, right?
Yeah, he made a detour to the Primate Center, and we got to hear his story.
Yeah, actually, he emailed me, and he said, I'm talking with Welkin or something. But anyway, I wanted to do a TWiV with him. He said, no, we submitted this, and the editor says I can't do any podcasts until it's published.
On his news story, yeah. You definitely have to have him back once that comes out. I
just found that frustrating that here this journal is holding him hostage. Of course, he submitted it to a very high-profile journal. He wants to get, a high profile, helps him get funded and all of that, and that's the problem that we have.
Well, but I can kind of understand that. I mean, we have ourselves complained about pre-release of stuff that hasn't been published in peer-reviewed journals yet.
Yeah, but TWiV is peer-reviewed, isn't it? You're right, Alan. I'm sorry. I'm glad you keep me honest here.
But it is aggravating because Jeremy talks about that work at meetings all the time. So if you were sitting there with your TWiV microphone.
No, that's meetings usually have stipulations about what can and can't be covered. And reporting results from a meeting is one thing, and there's a particular context for that, and it's well established. But if we had him on the show just talking free form about what he's working on, and he says something that's a little beyond what the published data or the data in his talk would say, then we can get into some trouble with that.
Oh, then he jeopardizes publication, right?
It jeopardizes publication, but the reason for that, it's not just a silly rule by the journals. It's because they want the peer-reviewed research to come out and be available for people to read and analyze rather than just some off-the-cuff comments from the researcher.
Right.
Makes sense.
Yeah, well, this happens in the XMRV field, as you know, that people are always talking before the stuff is published, and it gets confused and misinterpreted. And
Right.
Yeah. Okay. So when did you jump into the TRiM story, Welkin?
So it's kind of a personal story. So in 2004, Matt's paper came out and sort of solved one of the problems, figured out what the source of this monkey block was. And like I said, that sort of kicked open the floodgates. I counted once, in the first 3 years after that, there were over 100 peer-reviewed publications on TRiM5. So it became a really interesting but competitive field. And so at that time, I was actually sitting around waiting to find out if I was going to get my first faculty job. And that was a little frustrating because that was something I had wanted to work on, and I sort of felt like the world was passing me by. But I got my first job, I think, probably a year after Matt's paper came out. And one of the first postdocs, I hired 2 postdocs right away, and one of them was a woman named Ruchi Newman.
Where was that job?
Actually, I shouldn't have said my first job, my only job. It's where I am now. It just seems like a long time ago.
Right. Okay.
So Ruchi joined the lab, and she was kind of interested. She came from a bacterial pathogenesis background and was getting into virology, and was interested in the sort of the phenomenon of cross-species adaptation and restriction, and was interested in TRiM5. And she actually started working on a different project, but with regard to TRiM5, we thought, well, maybe one of the things we'll just do is pull some sequences out of rhesus macaques and see what it looks like. And across the hall from us at the time, there was a neuroscientist named Greg Miller, who was already doing sequencing lots of genes, brain genes. I don't know what they were, but he was doing a lot of sequencing from monkeys anyway. So he had a lot of DNA samples. So we randomly picked, I think, a dozen of those samples and amplified TRiM5 sequence out of those. So this was 12 animals. And what we found in those first 12 animals was really very striking. We found 5 different alleles of TRiM5. So 12 animals at maximum you would have 24 different alleles, right? But for almost any other gene, you'll only find one allele. Maybe 1% of the time you'll find another variant. So any housekeeping gene, most other genes you look at. And here, for this particular gene, in just the first 12 animals, we had found 5 different variations on the same gene. So that was kind of extraordinary. The only place normally you would see something like that would be immune system genes, things like MHC or antibody genes. So we didn't know what to make of it at first, but we knew it had to be interesting somehow.
So for the uninitiated, then what you mean is that these variations are a couple of amino acids different in this gene from animal to animal, right?
Yeah. So let me just back up. So probably less than a year before we started working on it, a couple of labs, in particular Harmeet Malik's lab, a postdoc in his lab named Sarah Sawyer, had pulled TRIM5 orthologs. So right after TRIM5, the story came out, they went and pulled TRIM5 sequences out of some 20 to 30 different primate species, including Old World, New World monkeys. And they did an alignment, and what Sarah saw that was really quite striking, if you see it, was that there was a very high degree of amino acid difference between species, much more than you would expect given the time frame of primate evolution. And so she was able to calculate that there was a very high rate of amino acid evolution going on in the TRIM5 gene.
But in doing that, she's just looking at a sequence from a single individual for each species, right?
Right. She was doing interspecies comparisons, yes.
Right.
And what she did that was kind of neat is she took it a step further. So she compared the sequences. She was able to argue that the sequences, the amino acids of the proteins were evolving quite quickly compared to other genes. And she noticed that all the variation tended to be very highly clustered in one or two parts of the protein in this C-terminal half called the SPRI domain. And so she hypothesized that those regions that were so variable were probably the regions that determine recognition for different retroviruses, right? The idea being that what's driving that rapid evolution in the SPRI domain would be the history of retroviral epidemics sweeping through our primate ancestors for the last 30 million years.
So this brings up something. Sorry, I hope this isn't too much of a distraction, but in your mind, is TRIM5, is that evolved specifically to deal with retroviruses, or does it have other functions, or or is it antiviral for other viruses as well?
I think it my feeling is it has evolved specifically to deal with retroviruses. It might have activity for other viruses. There's one paper from David Knipe's lab that suggests it might have some activity against herpes viruses. But I think whatever it does, it doesn't have some housekeeping role or some required cellular function.
Somebody
right?
must have made
it's an antiviral.
Somebody must have made a TRIM5 knockout mouse, right?
That's actually no, because they probably have, but it's not informative because as far as I know, no one has found a virus that the mouse TRIM5 inhibits. So there wouldn't be a way to ask that.
But the mice, basically the question is, are the mice okay?
Are the mice okay?
Yeah,
right.
Yeah. I'm actually not sure if that's been done yet or not.
Okay.
But what I can say is that dogs are a natural knockout of TRIM5.
Oh.
So if you actually, Sarah Sawyer, I think, first pointed this out. If you go to the dog genome, what they have there is a bit of sort of, it's been interrupted by some kind of insertion, and so you just sort of have the detritus of a former TRIM5 gene there.
Okay.
What about humans?
What's that, Dickson?
What about humans, us?
Humans have a TRIM5 gene. We have a functional TRIM5 gene. It's not very good against HIV-1, but it will restrict other viruses. So in the laboratory, human TRIM5 can work against mouse retroviruses, for example.
How about non-human primate retroviruses?
Okay.
Does it block them?
Does the human
TRIM block non-human SIVs, basically?
It depends on which strain you look at.
Yeah.
Yeah, and I can go into that a bit when I talk about.
I think it's remarkable that that TRiM may have evolved just for retroviruses. I don't know of any other kind of immune system product that would be so pathogen specific.
Well, I think of it as a pathogen recognition receptor, right? It's exploiting something that's uniquely retroviral, the same way a toll receptor, say recognizes LPS as being
and look at all the ERVs in the genome. Clearly,
I don't know
retroviruses have been a big factor in evolution.
Yeah, I guess that's it.
Beginning.
They've really assaulted us.
I don't know that it's really all that specific when you consider the magnitude of the threat.
Yeah, maybe.
That's actually a good way to put it. That's usually how I try to answer that. If you look, I think we talked about this the last time I was on, but if you look at the sheer number of ERVs, endogenous viral fossils in our genomes, and if you recognize that number is probably a very tiny percent of all the retroviruses that we've encountered, then you realize that things like the current HIV-1 epidemic have happened potentially hundreds or thousands of times during primate evolution. It's probably a regular feature of the evolutionary landscape.
Mm-hmm. But we have as far as recorded human history goes, we don't have any sense of some of those. The only one we know of is HIV, right?
Well, we have HTLV1.
Yeah, and contemporary ones, but old. We have Egyptian records which suggest that polio was around, 3,000 years BC, but there's no hint of any other retrovirus infection. We have hints of smallpox, right? You can see that in the literature, other viruses, but not retroviruses. You're right that it's a huge threat.
Well, there is modern data in the form of surveys. So when people, Walid Hainini and other people whose names I'm forgetting, when they do serosurveys in humans in areas where there's endemic retroviral infections of the local non-human primates, it looks like humans are actually exposed quite a bit to simian foamy viruses, simian type D viruses, simian lentiviruses. And so we actually may get hit with these things quite a bit. And it's sort of an open question why those don't always turn into epidemics.
Maybe because we've evolved such effective defenses against them.
Right. There's something that's kind of that's the direction of my life.
Maybe because these are such ancient viruses, before there were humans, we were evolving defenses against retroviruses, and now we've got it to a point where they're usually not going to end up succeeding.
That may be.
I don't know.
So there's a group.
Let's figure it out.
There's a bunch of people out there that are being surveyed for why they don't catch HIV. Have any of those been identified as having a special, different TRiP than everybody else, or TRiM rather?
Not really. So there's been 5 or 6 gene association studies. So studies where people have taken cohorts of HIV infected patients or cohorts of HIV exposed patients, and they've typed their TRiM5 alleles, and I think there have been some reports of weak associations between particular TRiM5 alleles and lower susceptibility to HIV-1, but nothing consistent from one study to another, right? So some of them have reported associations, but I don't think any 2 papers have reported the same association.
Uh-huh.
So there's some hints, but nothing relative to TRiM5. There's other genes that clearly have an effect. Right, that there's a large proportion of people in Europe that carry this deletion in the co-receptor for HIV-1, right, CCR5. So individuals that are homozygous for that co-receptor mutation are highly resistant to HIV-1 infection. But I don't know of anything other than those association studies. I don't know of anything else with TRiM5.
So this thing has evolved fast and is very polymorphic within a species.
Yeah, so then when we got into it, what we were seeing within a species was essentially the same thing that Sarah had been seeing between species. And so the explanation for that, we feel, has to be that in the rhesus macaques, the fact that they have all of these different alleles at high frequency must mean that something in their history has worked to preserve multiple alleles of the same gene. And we've argued before that it might be some sort of balancing selection. And I think if people have heard about balancing selection, you usually hear about it in terms of the sickle cell trait in Africa. How humans in regions that have endemic malaria there's a high prevalence of the sickle cell mutation in hemoglobin, and that happens because of the balance between needing normal hemoglobin to survive and the fact that the mutant hemoglobin confers resistance to malaria. So you have a balance of selective forces so that heterozygous individuals have a selective advantage, and so both alleles are sort of preserved at roughly the same frequency. So what we think might be going on in the macaques is a similar idea, but not just over thousands of years like the sickle cell trait, but over millions of years, encompassing the history of the macaque genus. And of course we can't prove it, but the idea would be that the assault on the ancestors of macaques by different retroviruses have effectively kept all of these alleles in circulation for a very long time.
What is the balance here, though? Because if I understand correctly, the main function of TRIM5 alpha is to prevent retrovirus infection. It doesn't have to balance a cellular function against that, right?
Right. No. So that gets to the functional work. So if we're going to argue that there's selection to maintain these alleles separately, then they must have some functional difference. Okay. Right. Because natural selection works on phenotype, right? It doesn't see sequence, and so we don't know what retroviruses were around 1 or 2 million years ago. But what we could do, and what Rucci did do, is she just took laboratory strains of different retroviruses. She engineered cell lines to express each of the rhesus monkey alleles, and then challenged those with different retroviruses. And there's clearly different patterns. So some of the alleles will block, say, retrovirus 1 but not retrovirus 2, and other alleles will block retrovirus 2 but not retrovirus 1, and some of them can work on both, and some of them don't work at all. So what it looks like is that the different alleles have slightly different specificities for retroviral capsids. So there's no one TRIM5 that recognizes every retrovirus. They're sort of tuned to particular capsids, and that probably reflects the history of any given species. The specificity of its TRIM5 has probably been molded by whatever retroviruses it saw in the past.
Well, so a better analogy than sickle cell might be MHC.
MHC is actually a better analogy. So the same phenomenon.
There's a selective pressure to have a diversity of these TRIM5s in the population, so that if a new retrovirus comes through, it will likely be inhibited by some subset of them, and it may kill a bunch of the population, but there will still be survivors to propagate the species.
Yes, that's exactly.
And that's actually a good point, Alan. I was sitting here thinking about individuals. You have to think about this in terms of population dynamics.
Right.
It makes sense to have a variety of alleles in the population, okay, so that if something new comes along, some people are going to some individuals are not going to make it, but the population survives because of the heterogeneity.
Right. Evolution operates on populations, and that's what
not
makes
on individuals. Right. Yep.
So that brings up an interesting point, actually. So what was really interesting about what we saw in TRIM5 is that we would see the same sets of alleles in other species of monkeys. So sooty mangabes and rhesus macaques actually share some of this polymorphism, meaning that multiple alleles were probably present in a common ancestor of the two species, and multiple alleles have survived through the last 10 million years in multiple speciation events. I'm not sure if I'm making that clear, but
that's clear.
So that sort of goes against the idea of genetic drift, where you expect over time one allele will just come to dominate by chance. And so the fact that we see the same sets of alleles in more than one species suggests that no one allele had the chance to dominate, that they were all sort of important at different times. And the reason the MHC is actually a better analogy is because, as far as I know, the only other place that's been documented in primates is actually in the MHC. So humans and chimpanzees actually have some of the same allelic lineages of MHC. Also because of long-term balancing selection, probably.
So what you mentioned that when Rucci did these experiments, you called them retrovirus 1, retrovirus 2. So what did she actually use? Different SIVs or different old world monkey viruses?
So originally our well-thought-out scientific strategy was to get any virus we could find. Mm-hmm. So we, because we work here, we had SIV MAC and HIV-1 and HIV-2. We got Mason-Pfizer monkey virus from Eric Hunter, so that's a different genus of retrovirus. And we got a couple gamma retroviruses from Jonathan Stoy, so those are mouse retroviruses. I forget, and foamy viruses. We got some spuma viruses too.
Okay.
So the idea was just we were looking for anything to distinguish the alleles from one another, so then we could have a handle to study them.
And also these are present in infecting old world monkeys, right? Old world primates.
The SIVs are, the mouse retroviruses
They do not, but the, how about, I'm looking at your paper here, you mentioned some gibbon ape, leukemia virus, baboon, endogenous viruses, and the foamy viruses. Are those all present in old world primates?
Yes, they are. Yeah. We haven't looked at many of those. Some of them are hard to track down functional clones of, but it would be nice to do that. So as a side note, one of the really interesting things about TRIM5 is that there are some homologs of it, certain species you can take their TRIM5 that will actually block infection by completely unrelated retroviruses. Mm-hmm. Right? So there are orthologs, for example, that can restrict HIV-1 and foamy viruses. So they're both retroviruses, but if you line up their capsid protein sequences, there's almost no identity. And so it means that whatever TRIM5 recognizes, it's probably some structural feature of capsid that the structure itself is highly conserved and uniquely retroviral.
So implied in what you're saying is that we don't really understand the details of the interaction between TRIM5 and capsid. Is that correct?
Right. That's true. Yeah, I think a lot of that still needs to be worked out. What actually is being recognized by TRIM5, and sort of as a corollary to that, what confers resistance to TRIM5 is when a virus is resistant, is it because it's not recognized, or is it because it's resistant to whatever the effects of TRIM5 are?
Couldn't you use two different fluorescent probes, one for TRIM5 and one for the virus, and find out where they meet in the cell?
Well, that sort of thing is known. So what's known is that TRIM5 encounters the virus in the cytoplasm. It even looks like it probably coats the outside of the viral capsid. Whether that's related to mechanism, I think, is still up in the air. But so it recognizes and coats it. The other thing that's known, I think this is still the case, is that TRIM5 doesn't seem to bind to monomers of capsid. What it recognizes is the higher order assemblies of capsid. So even though the capsid sequence determines whether or not you're recognized by TRIM5, it's when that capsid is assembled into the core that surrounds the genetic material. It's something about that TRIM5 actually seems to see.
And what happens subsequently? Is the capsid degraded in some way? We don't know.
I think what people think now is that TRIM5 binds and maybe triggers the premature disassembly of the core.
I see.
And that's important because that whole RNP sort of has to stay intact until reverse transcription is completed and the whole thing migrates to the nucleus. And so it may be that what it does is it sort of knocks that apart before it's had time to complete those early steps of replication.
So one of these days there's going to be a structure of TRIM5 interacting with a capsid.
Right.
Yes. Hopefully.
Crystallized protein.
Yep. So why don't humans have a good TRIM5? Is it just that the virus has not been in us long enough?
So Harmeet Malik and Sarah have argued, and they have a paper, I think it was a Current Biology paper a couple years ago, and they argue that probably what happened is that there was some sort of sweep a while back in human evolution. Perhaps some pathogen came in and selected very strongly for one particular version of TRIM5, one particular allele of TRIM5. And so we're not as variable as the monkeys. And that version that we got left with, just by chance, well, either by chance doesn't work against HIV-1, although I still think it's possible that HIV-1 also may have adapted to it early after getting into humans. I don't know if we know that for sure.
How broadly have the population analyses gone on looking at human TRIM5s? I'm just wondering if there are differences, say, in European versus Asian or African populations.
A little bit of that has been done, and there's some hints that there might be differences, but I don't think there's anything very strong. That's in the human cohorts. The only sort of really broad study I know is the one that Sarah and Harmeet did, where they basically took a human diversity panel and they cloned the TRIM5s out of those and looked at the sequences and did some functional assays in the lab. But I don't think they saw many differences.
Okay. I was just wondering, because when you talk about a pathogen sweeping through a population, I think about something like, bubonic plague.
Yeah.
But that was strongly regional. So,
well, if this happened, this could have happened. I don't know if they put a time point on it, but it might have been in the last 1 million or 2 million years. So
right.
I'm not
right.
sure where geographically it would have happened.
Right. Could have been before we even left Africa.
Yeah, and in fact, humans do have some polymorphism in TRIM5. It's just they don't have polymorphism in the spry domain, the part that recognizes the virus. There, it looks like most humans have exactly the same sequence.
So I suppose we haven't thought about making humans transgenic for monkey TRIM5, right?
I think of it in my mad scientist laboratories. I'm sure there's people thinking about that, and the other idea that I've heard, which I think might be a little more practical, a little more down to earth, is the idea of, because the human TRIM5 is functional, would it be possible to screen for and identify a small molecule
sure.
that would bridge it to HIV-1?
Yeah.
That would sort of make up, because it actually, even the human TRIM5, at least in our hands, does have some activity against HIV-1. It's just not very strong. So might there be a way to enhance that interaction? And I think I've heard Sidrowski and Jonathan Stoy and other people suggest that idea. So it's possible that people out there are trying that.
Is it easy in cell culture to select for viruses that are resistant?
That's been done a couple of times now. We haven't done it. I can think of at least 2 situations where people have done that. There's one report where MLV has been passaged in a cell line that was engineered to express TRIM5, and that acquired mutations in the capsid that escaped from TRIM5. Yeah. And then Sidrowski's group, I think, has done the same thing with HIV-1 in HeLa cells that were engineered to express rhesus TRIM5.
Yeah, so that's why you can't just put a functional protein in people. You get resistance.
The resistance can happen, yes. Yeah. Although, in vivo, it's a little different, right? Because there's probably going to be all kinds of synergy with other restriction factors and MHC and antibody. So it may be in the real world that TRIM5 is not this absolute block. It's not necessarily an absolute barrier. It's just a speed bump to slow the virus down. And if you combine the right things... my guess is in nature these genes really work more that way, that they lower the probability of infection. They're not resistance the way eliminating a receptor. The lack of something can really cause resistance, right? If you don't have a receptor for the virus, then you really are truly resistant. If you have a factor that acts against a virus, you're not really resistant because the virus can at least get in there in the cytoplasm in the first place to see TRIM5, right? And so there's probably some probability, if you're infecting enough cells, that some of those virions will get past the TRIM5 molecule and infect the cell, and the virus can start to replicate and adapt.
Well, you've already said you can overwhelm it by adding enough virus.
Yeah. Right? And you can, yes, that's true.
So what it means is that receptor selection by viruses commits the virus to a certain structure, and that it can't get around that if the receptor goes away.
Yeah. Yeah, if you're lacking something that the virus absolutely needs, I think that's the best, that's the most proven form of resistance that I know of.
So are you still doing this? Are you still working on TRIM5, or do you have a paper submitted so you can't talk about it?
No. So the most recent thing we did is, so after Rucci came to the lab, a student named Andrea Kermeyer joined the lab, and she really took on the project of asking whether or not TRIM5 really does in vivo what everybody assumes that it does, right? So almost everything else in the field, and again, it's hundreds of papers, suggests that TRIM5 works to prevent cross-species transmission of retrovirus. It prevents viruses from jumping into a population and starting to spread. And that's a combination of sequence analysis, like the stuff that Sarah Sawyer published that shows that the protein has evolved this way, and functional studies where people overexpress TRIM5 and it can make cells resistant. But what's sort of missing from that is in vivo studies to show that this is what TRIM5 really does. And the mouse homolog of TRIM5 is so different from primate TRIM5, and we don't know about viruses that it restricts, so there's no mouse model yet where you could ask that sort of question. And so what Andrea did is she went back to the story I started out with, this AIDS epidemic that was happening in the macaques in the 1970s. And she asked whether or not when SIV went from Pseudomangabey monkeys to rhesus macaques, however it happened, whether it was Gajacek or some other transmission, she asked whether or not the virus that emerged in rhesus macaques, SIV-MAC, whether it had to adapt to rhesus macaque TRIM5 in order to do that. And so one of the first things she did is she took Pseudomangabey isolates of SIV, so the relatives, the very close relatives of SIV-MAC, right out of the natural host of Pseudomangabey, and she took SIV-MAC, which is the macaque version of SIV, and she tested those in tissue culture. And what she found was that the macaque virus, the SIV that grew out in the 1970s, is resistant to all the different TRIM5 alleles that are present in rhesus macaques. But that the Pseudomangabey viruses, the progenitors, if you will, of SIV-MAC, are almost uniformly sensitive to about 2/3 of the alleles that are present in rhesus macaques. And so that suggests that when SIV from Pseudomangabeys jumped into macaques, initially it was sensitive. It could be blocked by rhesus TRIM5. And so what must have happened during the 1960s and 1970s is that the SIV that was starting to circulate in macaque colonies somehow adapted to overcome, or adapted to be able to replicate in the presence of the macaque alleles of TRIM5.
If you compare the sequences of those viruses, can you identify, are they very different, or are there sequence differences like in the capsid or something that would be appropriate for such an adaptation?
Yeah, and I don't want to reveal to the listeners that you're probably looking at my paper and leading me with that question.
Right. As a matter of fact, I have looked at your paper, but I'm just thinking. I'm thinking ahead.
It's the magic of TWiV. I've revealed the magic. So
pay no attention to that man behind the curtain.
The Chicanery of TWiV.
So Andrea, what really made the paper then was two things. One was she was able to look at alignments of the surface segment of the capsid protein from SIV, from Pseudomangabeys, and from SIV-MAC. And there was two differences. So they're very, very similar, but there were two differences that really stood out. One was a proline that's very highly conserved in all the primate lentiviruses except SIV-MAC, where that proline now, it's actually an LPA, so it's 3 amino acids that are highly conserved, and only in SIV-MAC have those changed, and they've changed to 2 glutamines. So there's been a deletion and a couple point mutations, so it's a very drastic change. And the other is a very conserved, I think it's an arginine to a serine change. And just because they were so unusual in the alignment, she went ahead and looked at those two. And so she took SIV-MAC and reverted either one of those back to the ancestral sequence, the sequence that's found in all other SIVs. Just making that single change, either one of those, made SIV-MAC sensitive now to rhesus TRIM5. Right, so it was proof in concept that the capsid had adapted to macaque TRIM5 alleles, and that those amino acids changes were probably involved in that adaptation.
Cool.
Does that make sense? So
absolutely.
So that was the first sort of exciting. It sort of started to complete the circle. And then what happened is there was a meeting here in Boston, I want to say a year and a half ago or so, and Andrea presented her data at that meeting. She had a 15-minute talk, and at the end of that talk, a scientist named Vanessa Hirsch, who works on SIV down at the NIH, came up to us and asked us if we'd be interested in genotyping a cohort of animals that she was working with. And what Vanessa had done maybe a decade or more ago is she had decided to derive a new animal model based on SIV. So instead of using the SIV MAC that had adapted to macaques throughout the 1970s, what Vanessa did is she took an SIV directly from pseudomangabes and she passaged that through 2 rhesus macaques. So to sort of kick it off and give it a chance to grow in macaques, she passaged it through 2, and the virus she pulled out she called SIV-SM E543 because the animal number was E543.
So and she was looking for a nice simple name for it, yeah.
Yeah. And that, so that viral clone is the one that she started to work with instead of SIV-MAC. The whole field sort of had agreed that we need other versions of the animal model for other reasons. So she'd been working with this for 10 years, and so what she had was a freezer full of samples from a cohort of animals that had been control animals in those experiments. They had infections with E543 virus, but they hadn't been vaccinated or treated in any other way. And what was really striking about that cohort of animals, there was like maybe 43 animals in that cohort, is that the viral replication was all over the map. Some animals had very high viremia, 10 to the 9th copies of viral RNA per mil of plasma, so they look like HIV patients, actually. And other animals where there was almost no detectable virus at all and everything in between. And so what she had always argued is that meant that there was some underlying genetic feature of macaques that was variable, and that was resulting in variable outcome when you infected the animals. And did I say that clearly?
Yes.
Yeah.
Yeah, I understand. So
I'm all over this. I am all over this. You got me on the edge of my chair.
Yeah.
So if you have to go to the bathroom. So what
were their TRIM5 genotypes? Come on.
Yeah.
So Vanessa and Samoy Goldstein, who works with her a few years ago, they looked at about half a dozen of these animals. And what they were able to show is if you took primary cells out of a naive animal before it had been infected, and you challenged those cells in tissue culture with SIV E543, they saw a range of things. So cells from some animals were resistant to the virus and others were sensitive to the virus and everything in between. And in fact, that tissue culture experiment could predict what would happen in the animal if you challenged the same animal with the virus. So if the cells were resistant to the virus, the animal was resistant. If the cells were sensitive, the animal could be infected. So
it's a cellular effect.
So it was a cellular effect, and it was not adaptive immunity or something like that. And so when fast forward 10 years, when she approached us after this meeting, what she was suggesting is that the variation that we were seeing in TRIM5 might explain the variation she was seeing in her macaque cohorts. And so, bless her heart, she was right. That really made the story for us. We genotyped those 43 animals, and we saw a very strong correlation between TRIM5 genotype and whether the animal had high viral levels or low viral levels. It worked out very nicely. The TRIM5 by itself could probably explain 2 to 3 log differences in animal viral loads.
What a great story.
Nice.
Neat.
And the genotypes corresponded exactly to the tissue culture experiments that Andrea had done, where she had seen that SIV-SM was sensitive to some alleles and not to others. So that tied together nicely. And then the icing on the cake was that in some of the animals that had low viral loads, after 10 or 20 weeks, in some of these, the virus suddenly came roaring back. Mm-hmm. So initially the virus had been suppressed, and suddenly it was growing out, and the animals had high levels of infection. And when Andrea went in and pulled the capsid sequences out of those animals and looked to see if the virus had adapted, it actually had some of exactly the same changes that she had seen in SIV-MAC from the 1970s. Right? So that sealed it for us, that TRIM5 had a biological effect in vivo, that it could suppress a virus during the process of cross-species transmission, and really argued what all the evolutionary biologists are saying is that for a virus to jump into a new species, one of the things it has to adapt to then is the TRIM5 variant that's present in those species.
Mm-hmm.
Wow.
As
Great stuff.
Cool.
Yeah. As a practical matter, it also turned out to be important because it turns out that the viruses that Vanessa developed, a lot of people are using for preclinical AIDS vaccine studies in the United States. And so suddenly now there's this concern that the effect of TRIM5 may be confounding the outcome of those vaccine studies.
Mm-hmm.
So they have to genotype each animal, basically, right?
Right. So that's what we've, I think, for the last 6 months now, that's what we've been doing. People have been shipping us samples, and we've been typing them, trying to figure out what's going, see if TRIM5 is actually going to be a problem for those studies.
Yep.
You know, when I talk about this, I tell people that it's a glass is half empty or glass is half full sort of situation, right? So for people doing the vaccine studies, they wish this would go away, right? This is a problem and it's a glass half empty. For people like me, though, what you're really interested in is the genetics of virus-host interactions.
Sure.
This is really an optimistic thing. That kind of variation means there's something there to discover, right?
Yeah.
And thankfully for us, Vanessa had been paying attention to that and gave us that opportunity, really, to connect what we were doing in vitro to something that was going on in vivo.
Well, you're on the right side of that thing because it's not going to go away. It's there. You're stuck with it.
Oh, can I say one other thing?
Yeah, sure.
Because somebody might get mad at me if I forget. We're not the only people who did this. So Norm Letvin's group has published a very similar study where they looked at a cohort of animals that didn't have Vanessa's virus. They actually had SIV-MAC, and this is the virus that is supposedly adapted to all the macaque TRIM5s. And amazingly enough, what Letvin also saw a difference in viral loads, right? So the TRIM5 alleles also affect SIV-MAC, just not that much, but they were able to detect it. And so what's interesting about that is that even a virus that's adapted to a host doesn't have to be completely resistant to TRIM5 in order to be pathogenic.
So one of my questions is, there are other restriction factors, ApoBec, tetherin.
Yeah. Yep.
Are there similar polymorphisms in those genes?
Yes.
Okay.
We've started looking at, in the rhesus macaque, both of those genes are almost as polymorphic as TRIM5. So plenty
it's
of more
going to get
projects.
really complicated.
Yeah. Actually, and what makes it even more complicated is all three of those genes are on different chromosomes. So TRIM5, ApoBec, and tetherin are in 3 different chromosomes. And so what that means is if you have 3 or 4 alleles of each of those genes, in every generation, you can mix and match those at random because they're not linked to one another. Right? So you can take the different alleles of TRIM5, different alleles of ApoBec, different alleles of tetherin, and mix them in all different combinations. And so if what we saw for TRIM5 is true for ApoBec and tetherin, if there really are 3 or 4 different functional alleles for each of those genes, then there are actually probably something on the order of 200 different composite genotypes in rhesus monkeys, just for those 3 genes that are involved in blocking retroviral replication. So when you start to consider the genes as a group, you're looking at diversity now that's sort of approaching things like the MHC and antibody. Not quite, but it's getting there.
And is there more to find, do you think, in terms of restriction?
Yes, there's definitely. The literature is sort of full of hints and suggestions that there's other kinds of blocks. And then there's also in the primate lentiviruses, HIV and the SIVs, there's a couple of genes, VPX and VPR, that people think probably counteract some sort of restriction, but as far as I know, no one has figured out what they're counteracting. So they're not counteracting TRIM5 or tetherin or ApoBec, so there must be something else there.
Great story.
Great story.
Very cool stuff.
I even got some of that.
I heard you snoring, Dickson.
Oh, you didn't.
All right, I'm just kidding. I like to pick on you.
You have Dickson locked in a vault there or something.
Sounds like it, doesn't it?
I'm sorry to say I don't have a nice headset to resonate properly here.
All right, Dickson.
We've actually sealed him into a corner of the Lincoln Tunnel.
I'll send you a headset, Dickson. I'll buy you one.
I've got to take the other one back.
All right, why don't we do a few emails? I've saved a whole bunch over the last 6 months for you, Welkin.
Uh-oh.
So I'm gonna go. Let's go to the one from Luke first, who says, I'm currently working as a technician in the biochemistry and molecular biophysics department at Columbia, having just received my BA here in the spring. First of all, I'd like to join the chorus of praises for your dedication to doing this podcast. I first heard about it this past spring when I took the virology class as an undergrad. Well, that must have been mine, huh?
Hey, Vince.
And I've always found it to be a very entertaining and informative discussion of a topic which I find very interesting. If I may, I'd like to ask if you could touch on 2 subjects that I have recently read about. The first one I think you've talked a little about before. That's the subject of CRISPRs. Specifically, I would love to hear a bit more about the mechanisms by which they help defend bacteria against phage and how they could have gotten into the genome. Perhaps these things are not very well understood yet, but even some speculation would be interesting. So we did a show on CRISPRs, but you want to hear some more, huh, Luke? Okay. We'll have to do that either here or on TWiV.
Yeah, maybe we could get some CRISPR freak on the show.
Yeah, CRISPR freak, I'm sure.
I know some CRISPR freaks. I can.
There you go.
Right.
Okay, good.
I think I have a drawer full of them around here. Oh, no.
All right, send me them, Welkin.
I will.
The second is an article in the July issue of PLOS Pathogens entitled Unexpected Inheritance, Multiple Integrations of Ancient Bornavirus and Ebola virus, Marburg virus sequences in vertebrate genomes. The authors find several sequences in vertebrates, including humans, that seem to have been integrated from viruses in the 2 virus families mentioned in the title. Not only are there long single-stranded RNA viral sequences integrated into a double-stranded DNA genome, but they seem to be conserved and even expressed in some cases. One big question that I have is regarding the mechanism by which a single-stranded RNA virus's genome could be integrated into the genome of its host. They go into a possible mechanism involving line elements in the paper, but I'm not sure if I fully understood the details. I believe bornaviruses go into the nucleus, which is unusual for an RNA virus, but does the same go for the other 2? Are these integrations a result of an error in a pathway, and if so, is it a viral or host pathway? I happened to read this article shortly after listening to your ASV episode, where one of your guests mentioned that there is a potential for using viral proteins as drugs, which could interfere with the formation of viable virus particles when a virus infects a cell. Could this be a case of nature already having used this idea? There are also some papers after this one which basically showed a piece of almost every virus in the genome, right, Welkin?
Yeah, I think what Luke is asking is so in the case of the bornaviruses and the retroviruses, it's easy to picture how they get into the DNA because those viruses have part of their replication cycles in the nucleus where the DNA is, right? So the bornavirus is an RNA virus, but if its RNA is in the nucleus, all it takes is a reverse transcription step to get it into the DNA. And so I think what he's asking is, how is it that RNA viruses that don't get into the nucleus, how are their genomes getting in there? Is that how you...
I think
that's how I read it.
Yeah, that's how I read it.
So I think actually the answer is fairly straightforward. So no one's proven this, but what they argue in those papers is that the RNA from those viruses being picked up by line elements. These are the retrotransposons that can take RNA, reverse transcribe it, and reintegrate it back into DNA. And the replication cycle of line elements, of retrotransposons, usually they make a protein, right? So their protein has to be translated in the cytoplasm. And so the reverse transcription complex of the line elements is assembled in the cytoplasm. So it can pick up, just like a retrovirus, it can have an RNA that it picks up in the cytoplasm and carry it back into the nucleus and reverse transcribe it. So I think that's probably what's going on, that these elements, for some reason, either randomly are packaged into line RNPs or there's something about their sequences that makes them attractive to the line apparatus. Mm-hmm.
And so from that point of view, to answer his question, it's an error in a cellular pathway, really, a host pathway. One of his questions was, are these integrations a result of an error in a pathway? And if so, is it a viral or a host pathway? Well, I suppose it could be both, really, Okay.
Is it an error in a pathway? It's just the line elements sort of doing what they do.
Right. The virus is there, minding its own business, replicating in the cytoplasm. The line element is having its reverse transcriptase translated in the cytoplasm, and the reverse transcriptase does its thing on the virus RNA, and then drags it back into the nucleus and says, "Say it followed me home. Can we keep it?"
Yeah.
Okay.
As Rich has said, those cells are pretty sloppy, you know.
Yeah.
There's no errors. It's just things that cells do.
Right.
Yeah,
Yeah.
I guess the other possibility could be in a dividing cell, sometimes the nuclear barrier can break down.
I was thinking the same
Another
thing.
thing.
Yeah.
It traps some RNA. You know, when you say that something replicates in the cytoplasm and not the nucleus or the other way around, in my experience, very few of these things are really absolutely black and white. It's gray, okay? And there are going to be mistakes made all the way along. So some RNA is going to get into the nucleus, perhaps by that pathway during cell division. When the nuclear membrane breaks down, it could get incorporated if it's lying around back in the nucleus. So either way works.
Life is a messy business.
Life is a messy, but I like the line element explanation, though. That's pretty cool.
Yeah, that's good. Then Dickson, he also asks, he says, although from my understanding it's not really relevant to human disease, could Dick talk about the liver fluke worm, Dicrocelium dendriticum on TWiP?
I bet he could.
Could you, Dickson?
That's another question. Well, Dicrocelium dendriticum has a very unusual life cycle, so I would be willing to do that, but we'll have to put that near the end of the first sequence of our talks.
All right. That's a
Are
deal.
there things that Dick can't talk about?
Yes.
No, nothing. He can talk
As a
about
matter of
everything.
fact, I meant to mention that I don't have papers on TRIM5, so I can't talk about them.
Well, you can ask questions, though.
I could. Yes, I could.
The next one is from Aaron, who writes, I recently developed a science obsession that led me to learning about viruses and ERVs. I found your show in the search, and though most of the terminology is unfamiliar to me, my progression through your Virology 101 podcast, as well as some serious web searching, is helping to bring me up to speed. I listened to your podcast on ERVs, and I've read numerous articles and scientific papers. So that must be the one you were on, Welkin. The podcast on ERVs. That's the only one we've done. In all of that, I've never heard a comprehensive description of how an ERV enters the germline. Okay. In those regards, I have some specific questions. I'll let you take it after each one, Welkin. 1. How does a retrovirus bypass the blood-testes barrier to reach germ cells? I understand that HIV does so by hiding in white blood cells. Are there any other retroviruses capable of doing this, i.e. they must be able to bind to CD4 receptors?
So actually, I think maybe we should read all the questions because I think there's really one good answer.
Good. 2. Of the 5 types of ERVs, alpha retroviruses, beta, gamma, lentes, and spumas, is it known that any of them have the correct surface proteins to bind to germ cell-specific binding sites? I read that the common sperm cell binding sites are heparan sulfate and mannose receptors. I'm not sure if those are the same for spermatogonia. 3. Once inside a germ cell, how does a retrovirus successfully integrate its DNA into the host DNA? I read some HIV studies where the sperm of deceased HIV patients was examined, as well as studies where HIV was introduced to sperm in vitro. In no case was the viral DNA inserted into the host chromosomes, even though the virus was found within the germ cells. One study suggested that a low hydrolytic activity in the cytosol of sperm prohibited viral uncoating. Could the tightly wound structure of sperm chromatin also hinder viral DNA insertion? 4. My guess is that a retrovirus would have more success inserting its DNA in a spermatogonia stem cell than in a developed sperm. How would the spermatogonia be able to survive such an infection? Wouldn't the viral infection and subsequent release of new viruses eventually destroy the stem cell? If it survived, would the host produce sperm cells with the retrovirus insert for the rest of the host's life, or would the host have to reproduce while actively infected with the retrovirus before the immune system wipes it out?
Maybe I could jump in here before the last. So the reason I wanted to read them all is because I actually love this letter. I've read this 5 or 6 times since you sent it to me, and I would love it. Like if a student came to you with this set of questions, it would tell you that student had really been thinking about it.
Absolutely.
Yeah.
The answer to the question was, would you like to work in my lab?
Yeah.
Write my next grant.
Although he does seem to have a rather one-sided view of the germline.
So that's what I was going to say. If this was my student, I would use the Socratic method, and I would say, why are you obsessed with sperm? And go away and think about that, and then come back. Because I think, I'm half kidding, but I think you ask that question, the person might go away and think about it a bit. And so part of the answer is that sperm is not the only way to get into the germline, right? So sperms is one side of the story. There's also oocytes, which they're not tightly wound. They're actually replicating cells, right?
They're a much better target if you want to put some DNA in, I would think.
Right.
So telling him to go back and think about this, would that be egging him on?
Dixon. My goodness.
I have to ask, do any of you guys listen to Car Talk?
Oh, yeah.
Yes.
We do.
Oh, yeah.
We do.
So my friend Jamie Hensey describes this show as Car Talk for virologists.
Excellent.
Outstanding.
That's very nice. I
We'll
like
take
that.
Actually, and Jamie has written to you guys, and she's mad because you refer to her as a he when you read her letter.
Uh-oh.
Anyway, so back to the
Sorry.
So the other thing that can happen is it doesn't have to get into a germline, into a sperm or an oocyte necessarily. You can imagine that maybe the easiest way for this to happen would be infection in a very early stage embryo. And there's actually experimental evidence for that in the mouse model. So what would happen there is you'd have the early, I don't know the term for it, but something that's still at, say, the 2 or 4 cell stage or the 8 cell stage, where those cells haven't differentiated into any tissue yet. And so if they got infected, and if any one of those got a provirus in it, and that particular cell became the progenitor of germline tissue in the grown individual, then that germline tissue would carry that provirus. Right, and so cells, sperm or oocytes made from that tissue would have the provirus. So I think it could also, it's probably most likely to happen at that stage. So in sexually reproducing organisms, it's probably in a pregnant female where that would occur.
At least in mice, right? The stem cells are resistant to, I think, HIV, right? Or some, not maybe. Yeah. I don't remember the virus, but there is resistance at the stem cell level. So that would make more sense, yeah.
The other part of his question is how can the individual tolerate this? So if you get this very early formation of an endogenous sequence, why doesn't that kill the cell and kill the individual? And I think there's a lot of answers to that. Retroviral replication is inherently error prone, and it may be that much of the time the provirus that lands in any given cell is defective in some way, so it might not be expressed. So therefore it's not toxic to the cell. It might have a deletion in it or something like that. The sequences themselves will get methylated, so even if they're intact, they're not expressed. They might land in some part of the chromosome that's sort of tightly bound up and not accessible to transcription factors. The other part of the story, of course, is sometimes they probably do get in and they do kill the individual of the cell. My guess is 99% of the time or more, when a new provirus gets into the germline, it never sees the light of day. So the thousands of them that we see in our genomes may be just those cases where it worked out.
So I think you said this last time. You said, there might be a person walking around right now with HIV in his or her germline. But it's a very rare. It's a very rare event, right? And you won't see the effects of that for thousands of years, probably.
Yeah, and even then it would have to be the case that the person survived. The provirus is not detrimental to the person in some way.
Aaron closes by saying, this is very funny, I appreciate any help you can lend. I'm wondering if I will have to listen to a podcast to hear my answers read to me, or if you will respond via email.
Well, I think I can answer that.
It's funny. It sounds like he doesn't want to listen to a podcast, right?
Will I have to?
I have to. Well, Aaron, I don't know if you're still listening, but there you go. I waited for the expert, because otherwise we wouldn't have the best possible answer. The next one is from Tim. A fan of your show, I would like to hear your comments upon reading this relatively recent publication in the Journal of Virology, Isolation of an Infectious Endogenous Retrovirus in a Proportion of Live Attenuated Vaccines for Pets.
Yeah, this is an interesting paper from Massimo Palmerini. Did you want me to take this one, or was
Absolutely.
somebody?
Go for it.
Absolutely.
What Massimo is actually, he's a scientist, but he was originally a veterinarian by training, so a lot of his work sort of ties into veterinary science. And what they did in this, it's a very short paper, and they went to look and found an endogenous cat virus called RD114 or RD114 is present in a lot of the vaccines that are currently used to vaccinate dogs and cats, probably because those vaccines are produced either in feline cell lines or cell lines that have been exposed to this virus. And so at some level, I think in a lot of these, they found this. And it raises the possibility that when we're vaccinating puppies and kittens, that we're introducing this endogenous feline virus into our pets. Although I think he does go on to say it's unclear yet if that's had any detrimental effect.
As a matter of fact, I think he says that he thinks that, well, there's certainly no evidence that it does. Okay,
Yeah.
It's probably not doing any harm.
So what do we look at for evidence of harm in this case?
Dead cats.
I mean, they don't live that long to begin with, right? I'm not sure it would have an effect on... I mean, I think you need to look for the virus in the animals that are immunized, right? Though he says, I think, that there's no evidence... Well, there's no evidence that this virus is actually replicating in pets. So I guess
one would expect that exposure to RD114 will be of little consequence to cats, given that RD114 is an ERV of this species.
Right.
Okay, so normally cats have this in their genome, and what, they don't produce it, or they do and it's of no consequence?
Yeah, I don't know if you could rule out a consequence. I mean, mice have active endogenous viruses, and they clearly can cause disease in mice. Mm-hmm. There's an interesting aside to this. He writes, millions of puppies are vaccinated annually worldwide, and that reminded me that, as far as I know, no one has ever described a retrovirus of dogs, which is just remarkably. It seems impossible that there isn't one, and it seems impossible that if there is one, we wouldn't have found it, because we've been living together with dogs for a very long time. Any other domestic animal that you name has multiple retroviruses: cats, cows, chickens, pigs, everything.
Oh.
So why don't dogs have a retrovirus?
Or wolves?
I don't know if that's been looked at. I don't know if
that's where the dog comes from.
Yeah, I don't know if it's a carnivore thing or a dog thing.
Dogs must have a really cool TRIM5.
I've got a 1990...
They don't have one.
Oh, that's right. They don't.
No.
Wow.
Got a 1995 paper from Journal of Comparative Pathology, retrovirus-like activity in an immunosuppressed dog.
Yeah. So
it's a pathology paper. I don't think they actually...
So there's a handful of studies like that where there's hints of something, but there's never been... I think that paper's probably 15 or 20 years old, right? Or is that a more recent one?
It's 95, so it's...
Yeah. So I've never seen any follow-up to anyone who's connected that and actually gotten a sequence or isolated a virus. And it may be, even humans... There are human cell lines that will produce ERV particles that are non-infectious. So it may be that occasionally people are seeing that sprout up in dogs.
Yeah, and I'm not seeing anything. I mean, it's a lot of references to that paper and then a couple of other one-shot deals like that.
No, I've asked veterinarians about this, and nobody... I was in Ohio last week, and I asked Mike Larimore, and he said he couldn't think of one. Everybody does the same thing. They mention those 4 or 5 papers where there's hints, but I don't think anyone's ever... I've never seen anything about a full-blown exogenous retrovirus.
There's probably not a lot of funding for following up on those.
My argument would be that if the dogs are somehow resistant to retroviruses, it would be good to know how they did that.
Absolutely.
Yeah,
absolutely.
But
Sure.
I don't know if that's true.
They got ERVs, right? Dogs must have ERVs.
If NIH is listening, I'm willing to do the work.
It's problematic. Money is problematic. They may be listening. I know some people at NIH do listen, but money is very tight.
They don't really have control over how much money is going in.
It's Congress, right?
Pretty much.
Congress should be listening to TWiV.
I doubt they do. He says at the end the methods currently employed to screen veterinary vaccines for retroviruses needs to be reassessed, and it would probably be better to make them without the retrovirus in them. I probably would agree with that.
Sure.
Yeah.
He also puts in a paragraph on XMRV. I wonder why. He's talking
For his
about
next grant?
XMRV, and it says been associated with human disease, has not been proven yet. And he says, interestingly, current methods used for screening human vaccines for retroviral contaminants include extremely sensitive PCR that are much more sensitive than conventional assays. Thus, contamination of human vaccines with XMRV would not pass undetected with the currently available technology. Although this may not be true for vaccines produced in previous decades.
I guess the point that he's making there is, here's this virus that was just recently discovered, and yet the tools that we're already using to test our vaccine quality would already have picked it up if it were contaminated in there.
Yeah.
There's another. Why don't we use something like that for animal vaccines too? I think is what he's getting at.
Yeah. Right.
The other thing here is that they're related. So RD114 and XMRV are both gamma retroviruses, and the cat virus is interesting because its envelope protein, as far as I know, can function on a lot of different cell types. So it has a broad host range, at least at the entry level.
Yeah.
So in theory, if your cat or your dog is carrying this, you might be able to pick it up too. Not trying to start another conspiracy. I'm just pointing
It's
that
interesting. It's interesting.
I'm trying to divert attention from XMRV.
Good luck.
I don't think you can do that. No. I think probably we should move on to our picks.
Great.
Is that a good idea, folks?
Sounds good to me.
Works for me.
Because we can always do these next week.
Yeah.
We need to have Welkin back again. This is too much fun.
We should add him to our regular team.
He's welcome.
Yeah. Oh, yeah.
Welkin is welcome anytime.
I love it. I'll give up my day job if you want.
We don't do that either. But
the pay's not great.
you can.
No.
I'll let you know whenever we do it, and you can always jump in every Friday at 2 p.m. That's it. Let's do some picks of the week. And Welkin, what do you have this week?
So my pick this week, and I checked, I don't think it's been chosen before, is a thing called SupraMap. And I learned about this because I met the guy who developed it at Ohio State University, Dave Janies. And if you go to the website, what SupraMap is it's a way to integrate phylogenetic data and geographic data. And so what they, if you go to look at the visual representations, it's very cool. What they have is phylogenetic trees, which are 2 dimensions, right? That's time and sequence genetic divergence, right? It's an evolutionary tree. And what they can do is they can connect that to geographical data. And so the visual form of this, there's movies on the website that are really cool, is the root of the tree is up in the atmosphere above the earth. That's the history. That's the ancestor. And the tips of the tree, the tips of the branches are actually, those are the modern sequences or the modern isolates, say, of a virus that we know of. And those tips of the tree are touching the surface of the earth in the spots where they were isolated or detected. And he's got a movie on there, so you can look at like a flu epidemic in those 3 dimensions, geographically as well as evolutionarily.
That's
Neat.
And it's
Go ahead.
It's a really neat tool. It's a tool that's there for people to use. So anybody that has sequence and geographical data can use this tool to ask different sorts of questions.
I watched the movie, but I couldn't find like a file where I could actually play with one of the maps that had been created. That would be fun. Does that exist anywhere?
I haven't dug around enough to see that. I had the benefit that I was sitting in his office and he was showing it to me. So
That's cool.
It is very cool.
I think, Dickson, you would like this, right?
Well, that's going to be one of my picks is the latest issue of Science Magazine, which is all about envisioning data.
Okay, why don't you go ahead?
I just did.
You always
based on I had an earlier pick in which I took the Edward Tufte books of envisioning quantitative information. Those are great. They're absolutely fantastic. We all have big optic lobes, and that's how we absorb material through our eyes. And it soaks into our brain in different ways, obviously. And science is hard-pressed to take all this complex data that we're generating with DNA chips and all kinds of proteomics and, shotgun approaches to surveying molecules that might interfere with various biochemical processes. You've got all this data, but how do you actually massage it into something that you can understand? And so this whole issue is about massaging data into visual complexes that have appeal, they're beautiful, and they tell a nice story. So I'm all in favor of things like that because it makes then conveying this information to novice groups like, say, for instance, the voting public that might determine the budget for the NIH, a lot easier to do than it would be to have them listen to our podcasts and have to struggle with TRiM5 thinking, you're right, it's a diet pill, it's a protein in the cell cytoplasm someplace that interferes with some viruses in monkeys. If they could see it, they could repeat it to somebody else. And so that's why I'm really a big fan of visualization of complex data.
And this is also, that coincided with science, and the National Science Foundation have a contest for best science images each year.
Yep.
So that issue, that February 11th issue of Science is part of that. They announce the winners of that, and then they also have a bunch of articles on visualizing data.
Yep.
Well, I think it's great, Dickson, but there's nothing wrong with listening to a good podcast.
No, it lets you use your imagination to imagine. You can visualize this data.
I understand.
It might be better not to see some podcasts.
That's right. They serve different purposes, but that's pretty neat. Rich, you're up next.
Right. As you might know, I read very widely. Okay, and that includes the comics religiously every morning. Okay, and so last Sunday, Doonesbury ran a strip.
Oh.
Believe it or not, that included a bit on Jenny McCarthy and the vaccine autism thing. And I don't have the decades it would take to describe all of the Doonesbury characters. But in this particular one, Boopsie, who is kind of a wannabe sex symbol and apparently ex-playmate, is complaining. She's saying she's disgusted by Jenny McCarthy. She's complaining to her husband. And she's disgusted because Jenny is the one, quote, who's popularized a bogus vaccine autism link pushing tiny fraudulent study that was debunked years ago. She's done real harm to preventative health care, not to mention the Playmate brand. Okay? We're not all public menaces, she says. So I just thought it was great that whole thing had really made it to me when it hit Doonesbury. So that's good.
Can I add something to that?
Sure.
Of course.
If you don't mind. I recently met someone who doesn't have their kids vaccinated, and I was asking her if she was worried about, that they weren't vaccinated, and she was explaining to me that there's homeopathic approaches that she takes to boost immunity or something. And I didn't argue with her, but when I was driving away in my car, I was thinking about that, and I was thinking that we're selling vaccines the wrong way. The vaccines are actually the best example ever of homeopathic medicine, right?
Absolutely. I thought of exactly the same thing.
Vanishingly small amounts of material, and you introduce it, and it enhances your natural. You can almost see the slogan, right? And it boosts your natural defenses or something.
Absolutely.
That's exactly what it was invented by Chinese peasants.
Yeah, no, that's right.
Yeah.
That's right.
It's probably the only proven homeopathic medication.
Yeah, and it's a darn good one.
Yeah, that's a good point.
Can I say darn on TWiV?
Yeah, you can.
Yes.
Say
Yeah,
that.
that's okay.
No problem.
If it's okay for Prairie Home Companion, we can do it.
Alan, what do you have?
I have, continuing with the vaccine theme, just so happens that this news just came out. The National Vaccine Plan, yes, there is one, was released in 1994, and it hadn't been updated since. It was just this plan about, how to promote vaccination and how to measure the outcomes of vaccination nationwide. And the Department of Health and Human Services has finally updated it. So there is now a 2010 update to the National Vaccine Plan. They just announced this thing is now going through its revision cycle. It's a government document, so the 2010 version hopefully will be done by 2012. And you can go online. Of course, the whole thing is open. You can read it. I believe there's there's an address here. You can comment on it if you have thoughts about how to proceed with this. And I assume those would be intelligent thoughts about how to proceed with this. And it's it's a large, detailed document, but worth reading if this is a topic that interests you. Or at least worth skimming, I should say.
Great. No, I didn't know there was a plan.
There is.
And it's good to know that they're thinking ahead.
Yeah.
Someone is.
Great.
Is there anything in there about making it easier to modify protocols? So vaccines, say, that been made in primary cells for decades and decades would be safer if they could be made in cell lines. But I seem to understand that that's hard to do.
Yeah,
because of regulations.
I haven't gotten through the whole thing and all of the appendices and everything, but there's, it's kind of an outline of all of the issues. And one of the goals is develop new and improved vaccines. And part of the whole discussion around that is how do you make it either easier for the corporate world to do this or, get government labs more involved in it one way or another. There's obviously there are obviously some roadblocks in the marketplace there.
Yeah, it is a regulatory issue.
Yeah.
It doesn't look like in the outline here, but it would be easier if you could make it like the flu vaccine still in the U.S. isn't in cells.
Right.
Although we may get there eventually, but it's been tough. All right, my pick is a New York Times article which summarized the U.S. Supreme Court decision just this week, rejecting a lawsuit that faulted a viral vaccine. So by the vote of 6-2, the Supreme Court said the vaccine manufacturers should be protected by the vaccine compensation law, which went into effect, I believe, in 1986. And therefore, there's no point in suing. I think that's what it means, but I'm not quite sure. I'm actually going to have a chat with a lawyer friend of mine who has been involved in vaccine litigation sometime next week. 10-minute chat. I'll record it, and we'll play it on TWiV next time.
Well, you can still go to the vaccine court. Right?
There is a vaccine court. So it says here, people injured by a vaccine that say they were improperly designed must rely solely on a compensation system created by the law and may not sue vaccine manufacturers. That was what the decision was.
Right.
So you can go to the vaccine court and they will decide whether you get compensated or not, right? But you can't sue them in a court of law saying, I want millions of dollars. Somehow I don't think this is going to make any difference, but I will ask a lawyer. And
if it had gone the other way, it probably would have made a difference.
Yeah, well,
yes.
That's apparently what I think Pfizer was worried that there are a lot of here. Wyeth, the defendant in the case, told the justices that some 5,000 pending petitions concerning autism-related injuries, and that it feared a crushing wave of litigation. And one of the judges said she discounted those doomsday predictions.
I was interested in the voting here because the dissenting judges were Sonia Sotomayor and Ruth Bader Ginsburg. I wouldn't have predicted that.
That's very interesting.
Why wouldn't you predict it? Aren't they the most liberal?
Yeah, well, okay, I guess it depends on how you figure liberal or conservative works out in this particular case. But.
Yeah, and I think, as with most legal cases, it turns more on fine points of law than on the broad scope of the thing. I mean, the majority's opinion, and I have to say, it's nice to see that the Roberts Court has done something that I actually agree with. But they're saying that the intent of the law was to make a deal where vaccine manufacturers would be protected from ordinary tort law in order to take on the risk of making vaccines. And this decision is intended to preserve that deal. And then the dissenting opinion is that, no, there should still be the option to sue if the vaccine court doesn't offer the proper remedy.
So from that point of view, that's a liberal opinion.
Yes.
Well, this Vaccine Compensation Act in the mid-'80s went into effect because a lot of companies were pulling out of vaccine manufacture due to lawsuits.
Yeah, I think what it was called the doomsday scenario there is empirically true. The reason we have so few vaccine manufacturers is that this is not a very profitable business to be in, and it's extremely risky because of lawsuits.
Yeah. So my lawyer friend Roger Yorgis works for a firm in D.C., and he's been involved in polio vaccine litigation for, 25 years or so. And I've worked with him, so he has a pretty good handle on this. So I'm hoping to talk to him and get his thoughts. So we'll see if we can do that next week. And that brings us to the end of another juicy TWiV. You can find us on iTunes or at the Zune Marketplace. And you can also stream us to your mobile device, an iPhone or an iPad or an Android device using the Microbe World app at microbeworld.org. Or you can go over to twiv.tv, play the episodes, download them, and read our show notes. And, of course, we have a fan page on Facebook, facebook.com/thisweekinvirology. You can always send us your questions and comments to twiv at twiv.tv, or you can try our new phone line, 908-312-0760, or what some individuals have been doing, have been posting comments on Facebook at our fan page. So you can do any of those things to get in touch with us. Welkin, thanks so much for joining us.
Oh, glad to be here.
And the invitation is open anytime you'd like to join us. You're welcome to come by. Welkin is at Harvard Medical School and Small Things Considered, where he makes an appearance every now and then. Alan Dove, thank you.
Always a pleasure.
Good to have you back. Alan Dove is at alandove.com.
Where I make an appearance every now and then.
Yes, you make pithy comments. Rich Condit is at the University of Florida. Thank you, Rich.
Quite welcome. Always a good time.
bit.ly/poxdoc. And Dickson Despommier is in Fort Lee, New Jersey.
Yep.
Good to hear from you, Dickson.
It's good to be heard from.
Where are you? At verticalfarm.com?
Yeah, that's one of them. And Racaniello.org is another.
And rupertmurdoch.com.
No, medicalecology.com.
Very good, Dickson. Thank you. And get that headset fixed.
Yes, sir.
I am Vincent Racaniello, and you can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.